Today, we're excited to share this virtual lecture with all of you titled Taking a Closer Look, Harmful Algal Blooms in the Indian River Lagoon. Today's topic will be presented by Dr. Malcolm McFarland, a research associate and lab manager at Florida Atlantic University Harbor Branch Oceanographic Institute. So without further ado, Dr. McFarland, let me stop sharing my screen and hand it over to you.
Okay. All right, can everybody see that?
Yes.
All right, great. Well, thanks for that introduction, Kate, and thanks for inviting me to come talk. Always happy to do so. As Kate said, I am a research scientist at Harbor Branch Oceanographic Institute, right on the Indian River Lagoon, and I am primarily a marine ecologist, and I study phytoplankton and harmful algal blooms, both locally and in other parts of the country and in other parts of the world. So today I'm going to talk to you about harmful algal blooms in the Indian River Lagoon. And in case anyone doesn't know or has never heard of this before, I'll just give a brief definition here. And generally what we're talking about with harmful algal blooms, or HABs, as we often abbreviate it to, are high concentrations of microscopic algae that grow in the water. And these algae are also termed phytoplankton, which means they are microscopic and they float and they drift with the currents in the water. So there are other types of harmful algal blooms. Some people consider things like sargassum, which is a macroalgae, a large algae, tends to wash up on the beaches around here in large numbers. Some people consider that a harmful algal bloom as well. Or there are some types of algae that grow on rocks in shallow freshwater environments, and overgrowth of those can sometimes be called harmful algal blooms as well. But what I'm focusing on, and what's most of a problem around here, is these microscopic algae that float in the water and are called phytoplankton. And this is a tremendously diverse array of organisms. So some of the pictures you see here on this slide are just a few of them taken through a light microscope or an electron microscope. And there's a huge level of taxonomic diversity here. It ranges from prokaryotic organisms to eukaryotic organisms, and just an incredible deep division, much larger than we typically see even with land plants. And a lot of these are very old, evolutionarily speaking, and they're really quite fascinating organisms. So I'm always a little bit hesitant to talk about HABs because I study phytoplankton, and I think they're really incredible organisms. And presenting them as harmful is somewhat misleading, I think. So I always like to make sure people understand that these phytoplankton are really essential components of the marine ecosystem. In coastal ecosystems, as well as the global oceans that we have, phytoplankton are really quite essential. And in the top left of this slide, you're seeing the green, which represents sort of the average the average abundance of these phytoplankton throughout the world's oceans. And they're really widespread, of course. They're really concentrated in certain parts of the ocean, and they, on a global scale, they are responsible for about half of the primary production that happens on Earth, which means they're responsible for about half of the oxygen that is produced on Earth and the oxygen that's in the atmosphere. So about half of the oxygen that we need ourselves to breathe actually comes from phytoplankton. And in fact, before the atmosphere was oxygenated, it was phytoplankton that were responsible for changing the composition of the atmosphere and making it oxygenated thus enabling larger life to evolve. So as I said before, this is a huge, diverse array of organisms. In the bottom left, you see some electron micrographs of what some of these species look like. And they're not just little round dots either. They are complex geometries. This particular image is showing a diatom which produces a glass shell. These glass shells are very intricately patterned. They have lots of different geometric features to them, and exactly how they do this is still largely a mystery. On the right-hand side and in the middle, what you're seeing is some plates of drawings of these organisms and some arranged shells of these diatoms. When the microscope was first invented, these types of organisms were the first things that people saw, and they were completely fascinated. Early microscopists and early natural scientists were completely fascinated with these organisms, and they were the subject of lots of different types of drawings and cataloging of different species. And then it became very popular at one point for them to actually arrange these diatom shells and other types of organisms onto these display slides. And so what scientists would actually do is using a single bristle from a hog's eyelash, they would place these diatom shells onto a microscope slide in an arranged pattern to make something that looked like what you're seeing in the middle of this slide here. So with that being said, I hope everyone realizes that these are really critical components of the ecosystem, and they're really fascinating organisms in their own right. But they do, some of them produce these harmful algal blooms, which can cause real problems in our coastal ecosystems, and it's a problem that seems to be getting worse. So what's happening with these blooms, as we call them? Well, basically, a harmful algal bloom is a result of an overgrowth of these types of organisms. So you have too much growth, leading to sort of an imbalanced ecosystem state. And there are three main factors that affect the growth of all types of phytoplankton, including HABs. One is light, another is temperature, and another is nutrients. So in a very simple sense, these organisms are basically plants. They need light to do photosynthesis and grow. They need nutrients, just like land plants need nitrogen and phosphorus and other components of fertilizer. They need these nutrients as well. And temperature is another big factor. Temperature, generally warmer temperatures promote faster growth. And I think it's really important to note that we are, as humans, we are really currently having a pretty dramatic effect on both the temperature through global warming, and we also have a big impact, especially in coastal ecosystems, on the amount of nutrients that are in the water. And a lot of this comes from runoff from things like fertilizer, septic systems leaking, and runoff from agricultural operations. So there's a variety of ways that these blooms can be harmful. The most direct way that they can be harmful is by producing toxins. A number of these species can actually metabolize these compounds that are toxic to humans and other animals. And that results in a lot of, when we find these things in the water, that can result in closure of beaches and local recreational waterways to avoid contact with these things. Another big problem that they can cause is anoxia. Basically, if you get really high concentrations of these cells in the water, either directly or after they die through the promotion of growth of bacteria, you can get a lot of consumption of the oxygen that's in the water. And if that oxygen level in the water gets really low, low enough, things like fish actually begin to suffocate and die. And so what you're seeing here in the bottom left is the results of a fish kill due to low oxygen. This was in one of the canals adjacent to part of the Indian River Lagoon. And these anoxic events can be really devastating and just cause a huge mess. This is all dead fish that you're seeing here in the bottom of the picture. In some cases, this can be very localized and not a huge problem. And in some cases, when it's really bad, this can happen over a really wide area. Another way they can be harmful, especially in the Indian River Lagoon, is they can cause really high water turbidity. So they can cause when you have high cell concentrations, the water gets very murky, like you're seeing in this middle picture here, that really brown water. That you might look at that and think, oh, there's lots of suspended sediment in that water, or soil, or dirt, basically. But really, this is a picture of a bloom of a type of algae, and it's all due to really high cell concentrations in the water. And what happens when the water is really turbid like this is no light can penetrate to the bottom. And that really kills off and prevents the growth of things like seagrasses. And of course, in the Indian River Lagoon, seagrasses are a really important source of food for things like manatees. So it's this sort of ecosystem chain reaction that results in mortality of higher trophic level organisms as a result of these algal blooms. And this is something that we're seeing happen right now. I don't know if anyone's heard in the news recently, but there has been some unusually high manatee mortalities in northern parts of the Indian River Lagoon. And this is probably related to lack of food due to ongoing harmful algal bloom problems. Another less common way that these organisms can be problematic is actual mechanical damage. So some of these organisms, and I mentioned earlier that some of these organisms called diatoms produce these glass shells. Some of these diatoms not only produce glass, as part of their glass shells, they will produce these what are called setae, these sort of bristles, or these hairs. And a lot of these, if you look at this picture on the right, they can actually have these sort of thorns on them, basically, very sharp appendages made of glass. And when these get into the gills of fish, they can cause a lot of irritation, and they can actually cause fish to suffocate and die and cause fish kills in that way. Some organisms also produce a lot of secretions, and these secretions, mucus-like secretions, can actually do things like destroy the waterproofing on the feathers of birds, which ultimately causes them to get hypothermia and die. And this has been a problem on the western coast of the US, off the coast of California at times. And finally, from a human perspective, none of this is really good for economics. So there can be some really damage to local economies as a result of these harmful algal blooms. As you can imagine, people who normally use these waterways for recreation are not going to want to be coming to the Indian River Lagoon to boat and fish if the water quality is really poor and there's lots of dead fish floating around. So this problem of HABs is really been something that has been increasing dramatically over the last 50 years or more even. This slide on the left shows the reported incidences of harmful algal blooms around 1972. And you can see there's a few around the coasts. It wasn't an unheard of phenomenon, but compared to what we're seeing now, which is shown on the right, we're just seeing this all over the place now. This is something that has really, really exploded over the last 50 years. It's becoming much more common. And some of this is probably can be attributed to people becoming more aware of these things and them being reported more often and being studied more often. But it definitely seems like there's a clear link to the eutrophication of coastal ecosystems by introduction of lots of anthropogenically derived nutrients, and also things like global warming that are having an impact here. and causing a real increase in the frequency and also the intensity of these harmful algal blooms. And if you look down here in the bottom right, Florida in particular is one of the most impacted states in the US. So we really have this as bad, if not worse, than anyone else in the US. So next, what I thought I would do is I would just show you a few of the organisms and that cause HABs that we have been studying in the Indian River Lagoon. And there are actually, there's a whole bunch of these. And I'm just going to show you the few most common ones that have really caused the most problems and have even shown up in national news media outlets because of their intensity over the last few years. So the first one is actually a prokaryotic organism, and it generally comes from freshwater environments. This is a cyanobacteria. It's called Microcystis aeruginosa, and you can see it in a light micrograph on the right here. The cells are actually very small, so this is actually a colony of a whole bunch of cells. The cells are these little small dots, and they aggregate into these big amorphous colonies. And this one causes a real problem because it produces a toxin called microcystin. It's actually a family of toxins. There's lots of different kinds, but they're all liver toxins. They are very toxic. They can be very toxic to your liver and in humans and in animals as well. And sort of the thing that makes this unique, and one of the reasons that it can really outcompete other algae in the ecosystem and really grow out of control, is because individual cells contain these gas vesicles or little pockets of gas within the cell. And this is an electron microscope image you're seeing in the middle. And these honeycomb-like structures are actually these gas vesicles. And what that allows it to do is it allows these colonies to float up to the surface, and they form this scum on the surface. And that allows them to gather a lot of light, and they also shade out other species that might be competing for that light. The other consequence of that concentration at the surface is that you can get these very toxic slicks, these very concentrated pockets of toxin, which can be very dangerous for people or animals when they're coming in contact with it. So this is actually, this organism is actually blooming on Lake Okeechobee pretty much annually. And it blooms to the extent that we can really easily see it in satellite imagery. So this is an image on the left of Lake Okeechobee, and this green that you're seeing sort of modeled surface, that's all that microcystis. And when the lake level gets too high, the U.S. Army Corps of Engineers releases that lake water through the St. Lucie Canal to the east or the Caloosahatchee River to the west. And in doing so, if the algae is bad in the lake, it can transport that algae to places like the St. Lucie River and the estuary, and it can really accumulate there and cause a big mess. And it has done this in recent years in the past. And so this is an image of what that slick looks like in the St. Lucie estuary, where it can get accumulated. And so this is a real this is of concern to people and who might want to use this for boating, or it's really dangerous for pets that might not. There were several instances, I believe, of people's dogs being poisoned because they drank this water. People didn't realize that this green slime is really quite toxic. So there are reasons to be concerned with this guy. Another organism that is much more common in the northern end of the lagoon and has been for a number of years, is this Oreoumbra lagunensis. And this is another really small cell. These are only a few microns wide. So it's really tiny, but it gets incredibly abundant. And this, it's actually an interesting question to consider where it came from, because it was previously thought to be an organism that was primarily found way out in the deep ocean, and somehow it made its way to this coastal ecosystem, and it managed to find a foothold, and it seems to be really happy there. And so this can get incredibly dense in portions of the Banana River and Mosquito Lagoon and up near Titusville in the northern Indian River Lagoon. And as far as we know, it is not toxic, so that's a good thing, but it has been responsible for the loss of a lot of seagrass because it grows really dense. It shades out all the light, so no light reaches the bottom. And it's remarkably persistent. It doesn't grow in cycles like many other algae do. It seems to be always there in really dense concentrations. And if it gets dense enough, it has been responsible for some really bad fish kills due to leading to anoxia. So this is some images of that. This one I showed you previously. This is that really brown turbid water that results from the high cell concentrations, and you can see it in the bottom of this aerial photograph here as well. That water is just really, really brown. And this is a fish kill that happened as a result a few years ago as a result of really high cell concentrations of this organism. So another harmful algal bloom that maybe everyone in Florida has heard of is the red tide. This is much more common on the West Coast. It's caused by an organism which is a type of dinoflagellate called Karenia brevis. And this is a image of Karenia brevis through the light microscope and an image through an electron microscope on the bottom here. This is another relatively small cell, but a little bit bigger than some of the previous ones I just talked about. So this one is also very toxic and is responsible pretty much annually for fish kills on the West Coast. Some years are much worse than others. This last year was has not been very bad, but previous years have been really, really bad. And this toxin that it produces, brevetoxin, is very toxic to things like fish at relatively low concentrations. It doesn't take much. It's a neurotoxin. It attacks a nervous system, but it's also a real strong respiratory irritant. So if you're along a beach where there are high concentrations in the water of this particular organism, you can often feel irritation in your throat and in your nose, and it causes you to cough. And it can be pretty bad and quite problematic for people who have asthma or have other respiratory problems. So this one is really quite toxic, resulting in fish kills because of that toxin. But it's generally confined to the coastal waters, just in the coastal ocean, and not really in the coastal lagoons, at least not on the East Coast. And we usually don't see this on the East Coast, but it has come over here every now and then. Seems to be like a once in 10-year phenomenon, maybe, if history is anything to go by. And it happened, I think, in 2018. And what happens is it usually grows off the coast of Tampa and Fort Myers. And if it gets really bad, it can get picked up by the Florida current, and then it move into the Gulf Stream, and it can get entrained within these eddies that's shed off the Gulf Stream, and those can cause it to stick around the East Coast here for a while. And that happened for a couple weeks several years ago. And there were significant fish kills along the east coast here, as well as along the west coast. So I think this is the last one that I'm going to show, but this one is also really interesting. This is another dinoflagellate. This is another type of algae, microscopic algae, that grows up in the northern parts of the lagoon mostly, although we've seen it all throughout the lagoon. But it can get most dense in the northern parts of the lagoon. It's another dinoflagellate, like the Corenia brevis. But this one, when you look at it under the light microscope or under the electron microscope, you can see it has this hard shell covering. And it has these plates, and unlike diatoms, which make their shell coverings out of glass, this guy makes its shell covering out of cellulose. So this is like wood, basically. So we have algae that live in glass houses and algae that live in wood houses. And this guy is also toxic. It produces a very potent neurotoxin called saxitoxin. And this is responsible for what's called paralytic shellfish poisoning in humans. And it is also motile. One of the reasons it does so well in the Indian River Lagoon is because it can swim up to the surface during the day to gather sunlight, and then swim down to deeper waters in the nighttime to accumulate nutrients. So sort of like the microcystis, which can stay at the surface, it can go up to the top and outcompete other species because it can do this vertical migration. And this blooms pretty much seasonally, almost every year, I would say. Some years are worse than others. And another really interesting feature about this particular type of algae is that it is actually bioluminescent. And people actually organize, and you can pay to take a bioluminescent kayak tour up in the northern Indian River Lagoon, or the Banana River, or the Mosquito Lagoon, depending on where this guy is blooming at that particular time. And this is really wild. I've yet to do it, but I've seen this type of bioluminescence in waters, and it can be really impressive. When they're disturbed by your kayak paddle or by a boat prop, they will let off this beautiful blue light. And it can be quite a bit when the cell concentrations in the water are really high. And this is something that gets added to people's bucket lists, and it's really great thing to go do and see. I think most of these people probably don't realize that this stuff is also incredibly toxic. Now, it's not usually a problem as long as you're not jumping in the water and drinking it, but it is something to be concerned of. We have measured the toxins produced by this organism in the water, and they do, in some cases, exceed what are recommended guidelines for recreational limits. So you wouldn't really want to be swimming in that water for sure. Oh, one more. This is a diatom. So this is one of those that produces a glass shell. And this is one that we find really throughout the lagoon. This is an interesting organism. Most diatoms actually are not problematic. They're not HAB formers, and they don't produce toxins. So this is a little unique, and this is in that this is a diatom that actually produces toxin. And the toxin that this diatom produces is called domoic acid, and it's responsible for what's called amnesic shellfish poisoning. So if you consume a lot of this toxin, it will actually lead to brain damage and memory loss, and in extreme cases, death. So it's a really interesting toxin. It's actually thought that this organism was the inspiration for Alfred Hitchcock's The Birds. That birds eating this toxin that had accumulated in fish resulted in their going a little crazy. And I believe that someone actually, a scientist actually looked into that and looked into tissue samples from birds around that time, and I think they did find traces of this toxin in birds from that when that movie, before that movie was made, not long before that movie was made. So this one is really widespread. It's really common. We find it all throughout the lagoon, but it's not usually toxic, thankfully, in the lagoon. We have measured very small amounts of this toxin, so we think it's out there producing toxin, but thankfully not in high concentrations. But the cells can be very abundant in the water. So it's actually one of the major components of the phytoplankton out there. So it's definitely something we wanna keep an eye on. And there are multiple species out there as well, and not all of them produce toxin. They may have different ecological niches where they are found. So a really interesting character with this one. And of course, there are many others. Those were the sort of the major ones that we see in the lagoon, and we like to keep an eye on. But there are actually lots of other species out there that could potentially form HABs. So some of the ones you see in this slide, like on the left, this is another type of dinoflagellate called Alexandrium. It also produces that very potent neurotoxin saxitoxin like the pyridinium does. This is another type of cyanobacteria called Lyngbia, which produces Lyngbia toxin, very potent toxin. This we've seen in parts of the lagoon. It's called Akashiwo sanguinea. It's one of those organisms that can produce a lot of these mucus secretions and has been associated with bird kills in other parts of the world. This particular organism called Dinophysis, another dinoflagellate. I don't know if you're catching on, these dinoflagellates can be really problematic often. They're really interesting organisms. This one produces a toxin, Dinophysis toxin, that can be responsible for diuretic shellfish poisoning, so it can cause gastrointestinal problems. And then we've seen blooms of things in the lagoon that we have no idea what they are. So just recently, this past summer and fall, we saw an incredible bloom in the northern Indian River Lagoon of this little tiny cell. We believe it's a type of cyanobacteria, but we really don't know what kind it is. They kind of look like little coffee beans. And these got incredibly concentrated, turned the water to this crazy electric green color, and lasted for months, and actually did result in some fish kills, presumably due to low oxygen levels. So that's maybe a good place to pause here and take any questions that you guys might have. Next, I was going to talk about sort of the things that I do as a researcher and the other researchers that I work with about what we're doing to study these organisms in the lagoon. But if you have any questions on what I've talked about so far, I guess now's the time to ask away.
Yeah, Dr. McFarland, we've actually had a few questions come in through the chat. Stanley asks, can non-toxic algae be released to eat up the nutrients so the harmful types don't have a food source?
That's a really interesting idea. So it's very difficult to, it can be very difficult to manage or control these ecosystems in that way, because so many factors influence how these different organisms grow, and when they're competing for nutrients and light amongst each other, which one is going to win is really hard to predict. But that being said, what a lot of people would like to see is a resurgence or higher concentrations of these macroalgae, things like seagrasses and caulerpa. And those may actually help, if we have more of those in the water, those may help to soak up some of those nutrients, make less available for the phytoplankton. And probably be better overall for the stability of the ecosystem as a whole. so there's been things like lots of effort to replant seagrass, which is great. But that is really tricky, because in most scenarios, when you have macroalgae competing with the phytoplankton is gonna win. They just are better at soaking up those nutrients faster, and the light as well. So good
All
question.
right. They're good at being phytoplankton.
Yeah.
Thank you very much. And then a question from Jan. Are there any benefits to having these algae?
Well, like I said in the beginning, these are definitely fundamental, important components of the ecosystem. So they keep the water oxygenated when they're at a healthy concentration. They're actually the base of the food chain in most of these ecosystems. So the phytoplankton are eaten by zooplankton, small crustaceans, things like copepods, and these are the primary source of food for small fish. And small fish are eaten by larger fish, and so on and so forth. So they really are the foundation for the ecosystem. So without the phytoplankton, you basically don't have anything else. So they are very critical.
Great, thank you. And then we also had a question submitted from our Facebook live stream. Cindy was asking, I think this is in reference to the various toxins. She asked, could activated charcoal or bentonite clay be taken immediately after exposure? Would that benefit someone who ingested these toxins maybe?
Possibly. I'm not a health expert, so I would be hesitant to to that question definitively. I'm not sure what kind of treatments they do for exposure to these toxins, but that could very well be something that is done. I don't know for sure.
And then maybe one last question before we start proceeding is, these were actually submitted in advance of today's lecture, and you touched on these briefly, but just in terms of specific species of algae that we should pay extra attention to in terms of our pets' exposure or our exposure.
Yeah, so particularly the Microcystis aeruginosa, for one, that I showed there, that forms these really dense green slicks on the top of the water. If you see something like that, you can't necessarily be 100% certain that it's a toxic algae, but common sense and best practice would be to just assume that it could very well be toxic and stay away from it. Don't go swimming in it. Don't let your pets swim in it. Keep your kids out of it. It's probably best to avoid that. The Microcystis aeruginosa can be is really obvious when it's there in high concentrations, and some others like the Pyridinium bahamense, the bioluminescent one, that can be really obvious too, because you can see really high concentrations of algae at the surface. It'll turn the water red or rust colored, and you can see it. And anytime you see something like that, it's best to be safe and probably avoid it. Other ones like the Pseudonychia that I talked about, they can be really hard to determine if there are high concentrations of those cells in the water. They don't form really obvious blooms necessarily. So that's why we need to do this continuous monitoring, really, to make sure we understand what's going on and who is out there, and if we need to be concerned.
Okay, and maybe we can just take a pause to see if anyone would like to unmute. And we will have more time at the end for some Q&A, just as a reminder. Tracy, I see you unmuting. Do you have a question?
Yes, I do. I was trying to type it and I was getting all befuddled. When you say glass, Dr. McFarland, are you meaning silica as the structure, as opposed to being some other?
Yes, yes, it's silica actually. Okay. And what the diatoms do is they precipitate silica within their cells to form this glass shell, basically. And you can imagine that this is probably a good thing to make a shell out of if you're a photosynthetic organism that needs to gather a lot of light. So
Right.
it's a hard covering that protects them, but at the same time, it lets that light in that they need for photosynthesis.
Thank you.
Okay, well, looks like we can pause till the end for some more questions. So I'll pass it back over to you, Dr. McFarland.
Okay. So in the next part of the talk here, I was just going to go through sort of what we're doing to study these organisms in the Indian River Lagoon, both in my lab and also in collaboration with other scientists at Harbor Branch here and even at other institutions. So the primary thing that we do is we go out and we collect samples. And this slide shows that process. Basically, Harbor Branch has several of these small research vessels, which we take full advantage of to get out pretty much as frequently as we can and collect samples from various locations in the lagoon. The water quality conditions and the concentrations of algae in the water are extremely dynamic in marine ecosystems in general, and especially in the Indian River Lagoon. They're constantly changing. So we really need to stay on top of this to get a good idea of who's out there and whether or not toxic blooms are happening. So they change very frequently. A lot of these organisms grow, they can divide twice a day. So the populations can double 2 times a day, which means a bloom can develop from practically nothing within a few days if growth conditions are optimal. So we try and get out there as frequently as we can to sample water and bring it back for analysis. And the other tricky component of this is that the lagoon is a very diverse environment. And there are lots, it doesn't all behave the same way. So you can have a bloom of a potentially toxic algae in one small cove and you know, basically half a mile away, you can have a completely different situation in another cove where there's no problem whatsoever. So some of these blooms can be very localized, which means they're probably not a widespread problem, but in that particular location, they could be a big problem if they're producing toxins or they're consuming all the oxygen in the water. You can have a localized fish kill. So we get out on the boats and we collect a lot of samples. We collect samples in a variety of ways. Just collecting water in bottles, filtering it, collecting algae on filters, or using nets. And we also, while we're out there, we have a variety of different types of instruments that we use to measure water quality. So we will measure the chlorophyll that's in the water using fluorescence. We'll measure, of course, things like temperature and salinity. These are big factors that can affect how different algae grow. And we'll also measure things like the color of the water using various types of instruments. And I'll talk maybe a little bit more about that later, why that could be important. So, but usually we'll collect these water samples and we bring them back into the lab. We will look at them under the light microscope. And on the left here, you can see our light microscope in the lab. And this is what we see when we look through it at a water sample. There are a whole different variety of species out there. Not all of them are harmful. So we try and keep tabs on who's out there, but really focus in on these harmful species if we see them. And we can look at the organisms under the scope and count them. So we get actually concentrations of cells in the water, which is really important. So we have data to go on and keep track of. We also use an instrument called a flow cytometer in the lab. And this sort of automates this cell counting process. This is a flow cytometer is a type of instrument that was originally developed for medical purposes, to do things like count blood cells and make that process a lot more efficient. But we can use it for phytoplankton cells as well. So what it does is it draws in cells from a sample through a little intake tube, and it draws them in and passes them through a laser, basically in a single file line. So one at a time, you get these pulses of light as these cells pass through the laser. And based on how much light they scatter as they go through the laser and the colors that they fluoresce, we can classify them into different groups. We can also count them, and this automates the process. This is really good for looking at those really small cells, things like that Oreoumbra lagunensis, which is really hard to count under the microscope because it's so tiny. But with an instrument like this flow cytometer, we can do it really fast and efficiently and accurately. Then we also sometimes use some of these more fancy new instruments. This one is called a flow cam, and this sort of combines the microscope and the flow cytometer into one instrument. So it's sucking in phytoplankton from a sample, and it's actually taking pictures of them as they go through. So you get thousands of pictures of all these different types of phytoplankton, and you have this record of what was there in that particular sample. And we can go back and we can look for harmful species. We can determine how concentrated they were. So we go through that process and we collect all this data, and what we can look at is sort of how these organisms vary spatially and temporally throughout the lagoon. So for one of our projects, we are looking at these 5 stations in the southern half of the lagoon. And at these 5 locations, we go out basically once every 2 weeks. We collect the water sample, we bring it back to the lab, we analyze it in those ways that I just talked about, and we get cell concentrations. And we have, so on the top here, I'm showing total cell concentrations, is total number of phytoplankton that are in the water at these 5 different stations. Well, somehow I got an extra piece of data in there. Should be 5 lines, but instead it looks like 6. And we can also sort of zoom in or target just one particular HAB species with this data. So here on the bottom, I'm showing just Pseudonychia over time. So this is a time series for the data we've collected so far for this project. And this goes all the way back to October of 2018, and it's still ongoing. So we're trying to keep a record of all these different species that we're seeing, including the harmful species, at these 5 stations over time. And you can see for something like this Pseudonychia, back in 2018 and 2019, we saw some really intense blooms of this guy, and it since has improved recently. So maybe things are getting better with respect to this guy. We also have some other projects where we try and look at spatial distributions over broader scales. So in this project, we were looking at the lagoon-wide distribution of some of these harmful algal species. And this is that Oreoumbra that I was telling you about. This is the one that forms the really murky water in the northern parts of the lagoon. And we tried to measure its concentration all over the lagoon. And as we expected, we found it most concentrated up in the Banana River Lagoon and near there, and very low cell concentrations elsewhere. So the size of these circles on this plot is supposed to be indicative of the concentrations of the cells in the water. So we try and look at both where they are in space and when they occur in time for all these different types of species that we know are out there. And then we can combine that data, that biological data, with the environmental conditions that are going on out there in the lagoon. And for a lot of that, we use the Indian River Lagoon Observatory Network, which is a network of in situ sensors. It's run here from here at Harbor Branch by one of our professors, Dennis Hanasak. He's been responsible for setting this all up and keeping it going, and it's really fantastic. All this data is publicly available, and it includes things like temperature and salinity and the turbidity of the water, even concentrations of various nutrients that are important for the growth of algae. And then things like chlorophyll concentrations in the water, which is really an estimate of the total amount of algae that's in the water. And these instruments are out there at 10 different locations currently. And they're continuously measuring these water quality parameters every hour, basically. So every hour of every day, 7 days a week, 365 days a year, they are out there making these measurements, and we can go back and we can download that data, and we can compare that to our cell concentration data and what we're seeing when we analyze water samples, and we can really get a really comprehensive picture of what's going on out there in the lagoon and what may be causing these harmful blooms, what may be leading to their development, what happens when they die, what promotes growth of certain species versus other species. So this is all really important data and really critical to understanding the system as a whole and figuring out how best to deal with these harmful algal blooms. So we're also measuring the toxins directly. We use a technique called ELISA. In our lab, there are other ways you can do this, but ELISA has been working for us lately. This is enzyme-linked immunofluorescent assays. We can target the specific toxins like saxitoxin, which is made by the pyridinium, the domoic acid, which is made by the pseudonychia, and the microcystin, which is made by the Microcystis aeruginosa. And then again, we can look at the distribution of these toxins throughout the lagoon. And the circle sizes here represent the concentrations in the water of the different toxins. Something like the Microcystis, we're finding it down here, only down here in the St. Lucie estuary, really, and that's not surprising because mostly it comes from Lake Okeechobee. And the saxitoxin, we find it up here in parts of the Banana River Lagoon, which is where we find the pyridinium, so that makes sense. But if you look at this domoic acid, these circles actually represent low concentrations, but we're finding it really all throughout the lagoon. And studies that have looked at this toxin in animals like sea turtles and sharks have actually also been finding this toxin in these higher trophic level organisms. So it's getting into the food chain, it seems like. We're also looking at human exposure. This is some work that I did in conjunction with Adam Schaefer, who's an epidemiologist. This is looking at that microcystin in the environment in Lake Okeechobee and St. Lucie estuary, and at the same time looking to measure microcystin in the nasal passages of people who live in and around where these blooms were found. And what we found here in this study was that we were actually able to measure microcystin in people's nasal mucosa, and it did seem to correlate with the decline of the bloom in that area. So we didn't capture the beginning of the bloom, the before time, but we sort of caught the peak of the bloom, and then as the bloom went away, so did the concentrations of microcystin in people's noses. So this also suggests that it's not only in the water, but it's getting into the air. That it's getting volatilized, as they say, and people are actually who are in close proximity to the water and the blooms are actually breathing this stuff in. Now, these are very low concentrations, so we don't even know if this is a real problem, but it suggests that people are getting exposed, and it's something to think about. We're also using some various molecular genetic techniques to look at these. This is not so much what I do, but I collaborate with people whose specialty this is. We're looking for the genes of the organisms in the water and looking to see if we can look at all the DNA that's in the water and pick out these harmful species, and also looking specifically for the genes of the toxins themselves. So like I said, when we're out there, we're taking a lot of environmental measurements, and one of the things we're measuring is the color of the water. And the reason we do that is because the color of the water is basically what we can measure with satellite imagery. And if we can understand how the algae affect the color of the water, we can use these satellites to look and see where different algae are in the water. So on the left, you see some of these instruments that we're using to measure the color of the water. In the middle is another instrument that this one we have installed on Lake Okeechobee. This measures the color of the water. And you can see underneath it, there's a bloom of this microcystis, which is turning the water bright green. And then on the right, you see an example satellite image showing the southern half of the Indian River Lagoon. And you can see this nice blue ocean water and this really brown, dark lagoon water. You can see how those two interact and how the lagoon water comes out the inlets and can spread along the coast as well, which is really fascinating. So using these types of images in the lagoon is really challenging because you have a lot of issues with like bottom reflectance and lots of land effects and things like that. And it's only recently become possible that we can look at these coastal environments with satellite imagery because the satellite image data has gotten much more higher resolution. And so now we can really zoom in and see like these small tributaries and relatively small bodies of water that we weren't able to see before. So that's an active area of research. And then one thing that I've been involved with a lot and is really, really cool to me is we've been developing these submersible microscopes that we can put in the water and we can actually visualize and see the different algae directly with this microscope. And this thing can be installed and it has on board a little programmable computer, so we can put it out there, we can leave it out there, and it can actually monitor the algal concentrations. And we don't have to be out there all the time collecting samples. We can just look at the data from this and see what's out there. And so this is kind of what it looks like on the bottom left here. One side contains a light source and the other side contains a camera, and we can visualize all the different algae that are between the two windows here in the two housings. And this is what we can see. We can see things like this. This is a chain of diatom cells. This is another type of diatom cells that we can see. So this is still a work in progress. We're still developing this with some research grants that we've had for a while, and looks really promising. Really, really cool to see. So that's basically all I had. Of course, there's tons of other people that are involved in this research, and these I hope these are most of them. And we have funding from a variety of sources that really helps with this, and it's really a lot of different projects that come together, but all sort of focused on understanding the lagoon and the problems with harmful algae, and developing new ways to look at this stuff and figuring out how best to deal with it. And so I think that's all I've got. And if there's more questions, I'd be happy to take some more.
Wonderful. Thank you so much, Dr. McFarland. That was really interesting. I loved all the different photos and graphs. It's good to understand that just because you can't see it doesn't mean that it's not mighty, and it does impact us in so many different ways. Yeah. I do want to acknowledge that we had so much great information to share. We are at the 3 o'clock time limit. So if anyone does need to hop off for maybe another previous commitment, again, this is being recorded and live streamed. So you can find this recording at our Facebook page, the Nature Conservancy in Florida. Also, if you registered via Zoom, this will be sent out if you had opted in for that. However, if you are able to stay with us just a little bit longer, we are here to answer questions that you may have. So I'll go ahead and pass this over to Kristen to take any of those. You're welcome to unmute yourself and ask, or type that in the chat, and then we'll ask it for you.
Great. Well, maybe we can switch it up a little bit this time and see if anybody would like to unmute to ask their question before we answer the questions in the chat. Okay, well, I'll dive into the chat questions, and then if you do have questions, again, feel free to unmute yourself in just a moment. Dr. McFarland, we had a question from Margaret who asks, how do the Nature Conservancy and FAU collaborate with other environmental organizations who are concerned with the health of the Indian River Lagoon, such as the Marine Resources Council and the Environmental Learning Center, in addressing water quality and toxic bloom issues? So if you'd like to jump in, I'd be happy to provide a brief TNC summary when you're done.
That'd be good. Yeah, I can't speak too much to TNC, but I'd love to hear from you guys about that. We work mostly, I work mostly with some of the water management districts and the St. Johns River Water Management District and the South Florida Water Management District as well. So those are who we, as researchers here at Harbor Branch, I think are mostly working with, and also with state agencies, the Florida Fish and Wildlife Commission, the Research Institute, Florida Fish and Wildlife Research Institute. We have some projects that we collaborate with them on. So those are really the organizations that we most closely work with and have been either associated with through direct funding or just through a sharing of resources and ideas. And some of the other organizations you have been probably more indirect. I haven't had too much interaction with the Environmental Learning Center, although I am aware of them and have done a little bit there. And then we have like the here, not organized by myself, but by Dennis Hanasak again, we have the Indian River Lagoon Symposium, which involves a lot of other those organizations that are looking at issues in the lagoon from all sorts of perspectives. We also work with the Indian River Lagoon National Estuary Program. They're a big one that is really interested in water quality and blooms in particular, and so we do a lot of work with them as well.
Yeah, and I can just briefly for TNC, and Margaret, I'd be happy to provide more information also after today. We can always reach out to you, but TNC has a few different arms that all work within the Indian River Lagoon region. Firstly is our Marine Program Manager, Ann Birch. She's actually located up in the Melbourne area, and she participates on the Indian River Lagoon STEM Advisory Committee. So along with many of the agencies and NGOs that Dr. McFarland referenced, kind of being part of that collaborative community. And then, participating like in the Indian River Lagoon Symposium, like Dr. McFarland mentioned up at FAU. And I know Kate and I have many education and outreach partners along the length of the lagoon. Kate used to work for a long time in Melbourne, and I worked for a long time in Fort Pierce. So we're very fortunate to still be part of that lagoon community down here in Jupiter. All right, and then Dr. McFarland, we did have another question from Stanley, who asked, he mentioned that Sarasota has built some filtering trenches in Bayfront Park to try to stop runoff nutrients. Are you looking at any of these nutrient mitigation systems' effectiveness?
Personally, that hasn't been something that I've been directly involved in, but I am well aware of that is a really critical issue, and that a lot of people are looking at that. Some of the other people that I collaborate with here at Harbor Branch, like Brian LaPointe, has been really focused on the nutrient problem and the nutrients getting into the lagoon as being a real critical issue in fueling HABs. There are lots of ideas for mitigation. I'm not quite up on all of them, but I know that things like septic to sewer conversions are really a primary focus, getting people off of leaky septic systems, especially aging systems that are really probably responsible for a lot of nutrients entering the lagoon. But I do know there are other strategies I think that people are looking at, but not my direct area of expertise, I guess.
Yeah, it's such a multi-pronged issue. You know, you can really dive into, like, TNC also has a lot of freshwater work. We look a lot at nutrients in agriculture, in addition to all of these that all feed in. It's not a silver bullet type of solution. So
yeah,
it takes a lot of folks working in a lot of different areas to
it
try to
really
help.
It's a very complicated issue, and nutrients are coming from many different sources. And I think we don't really yet have a really good handle on which sources are the biggest problem and where we should most focus our efforts. And it may be different for different parts of the lagoon, and how we do that is really tricky. And it seems to me like it really requires a state level involvement, and it's not necessarily just going to be solved by individuals or local agencies, because it's too big of a problem, and it's really statewide. If you look at high concentrations of nutrients in Lake Okeechobee, those nutrients are coming from as far north as Orlando. Even the watershed is quite large, and then those nutrients end up in the southern half of the lagoon when water is dumped from the lake. So really big problem.
Yeah, and that is maybe a good segue into our next question, because I think, you know, the silver lining with so many of the severe issues we've seen over the past, you know, several years is that the public has become increasingly aware and really wants to help. So we did have a question from Charles on Facebook, who asked, what can be done to save the lagoon system, and what can the public do to help?
Yeah, well, on an individual level, you know, I think if you're a property owner, being aware of and being conscious of how you can limit what is coming directly from your property and going into the lagoon. Things like, you know, leaky septic systems are a problem, and applying too much fertilizer at the wrong time of the year. You know, that those are things that individuals, I think, can do that can make at least a small impact. And if enough people do that, that can be a big impact. But I think, you know, ultimately what's really required here is state level policy. That's the only thing that's really going to have a big fix. So, you know, I think it's incumbent on citizens and people to vote for the individuals that are going to make that happen. And it's a tricky problem, but we still need to know more in order to know what the right policies are. We need to know where those primary sources of nutrients are coming from, and how do we limit them while causing the least amount of, you know, economic hardship for people involved.
All right, well, maybe before we wrap up, we can take one last pause to see if anyone would like to unmute and ask a question. Yes, Mark.
Is there any way to get like a baseline on this whole system, like before humans started to make a mess? And because you wonder what was the natural interaction between all these different organisms before humans started building canals and drainage ditches and all that in South Florida?
Yeah, that's
any way to get a handle on that?
Yeah, it's a really good question, and it's a hard one to come up with a really good answer to, because back before these issues started to become a real problem, people weren't really collecting solid data and the types of data that we would need to really answer that question. But that being said, there's lots of historical records that seem to suggest, you know, waters in the lagoon 75 years ago, 50 years ago, were maybe even more recently than that, were mostly clear. The water was clear. The bottoms of the lagoon were sandy, not muddy like they are now. and that it was really a very different environment. And I think what has happened over the decades, a lot of the wetland landscape of Florida was changed, and they made all these canals to drain swampland, basically, and those canals end up transporting a lot of water very quickly into the lagoon, which was not the natural hydrology at the, that had evolved. And so that transports those nutrients very quickly into the lagoon. It also transports a lot of sediment and a lot of other muck. And those have very dramatically altered the environment in the lagoon. So getting it back to what it was originally may be very difficult, to be honest. But hopefully we can at least limit the damage that's occurred so far, and figure out how to manage this and manage the system better.